Hello students, I welcome you to EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am Senior Scientist in the Division of Animal Biochemistry at National Dairy Research Institute, that is NDRI, Karnal in Haryana, India. Today, I am going to talk about protein structure modeling from the paper Biostatistics and Bioinformatics. The function of all the biomolecules is determined by their three-dimensional structures. Proteins are macromolecules, biomolecules, and their function is also determined through three-dimensional structures, and the three-dimensional structures of the proteins is determined using experimental methods such as X-ray crystallography or NMR spectroscopy. Once these structures are determined, they are stored in the databases from which we can download and then use for modeling the processes. The importance of protein structures can be understood from the angle that unless until we have a three-dimensional structure of a protein, we cannot see its active site. Let's say this is an enzyme. So we will not be able to see its active site. Whereas, let us say this is a hormone receptor, then we will not be able to see its binding site for the hormone. So we need the three-dimensional structure. Let's say this is a transcription factor, again having a binding site for the DNA. So once we don't see the three-dimensional structure of a protein, then we will not be able to see the active site or the binding site. Therefore, the structure availability is must for understanding the biological function or the biochemical function at the molecular level. Therefore, in case the structure is not available, the absence, that is the absence of structure information is there, then this will not be able to reveal the function of the biomolecule. At the present, approximately 20% structures are available for well-characterized protein sequences. That is, for the rest of the 80% well-characterized sequences, we don't have structural information. Therefore, we are unable to understand their biological function to the full extent. Dear students, there are proteins which are homologous proteins because they descend from a common ancestor. Once they descend from a common ancestor, they have almost similar sequences which may be performing same, similar or even altered function. Because they have similar sequences, they will be folding into similar three-dimensional folds. So, the three-dimensional structure for the homologous proteins are almost similar. So, if we have the structure of one homologous protein in a group of homologous proteins, then that structure can act as a template structure from which we can borrow the structure and apply onto the sequence of the protein in hand for which the structure is not known. So, since we know that there is a relationship between the sequence and the structure due to the descent from a common ancestor and due to the similar folding pattern, we can borrow that folding pattern for folding a sequence in hand for which the structural information is not known. There are approximately 3,000 folding patterns, that is the folds which are there in the protein structure databases. Although the structures available are around, in May 2016, it was around 118,000 structures, whereas the known well-characterized sequences were around 550,000 in Swiss prot component of the UniProt knowledge base. Therefore, once we have concluded that there will be similarity in the structure due to descent from a common ancestor, and in case we are able to infer homology, then we can borrow the structural knowledge of the known protein and apply onto a homologous protein so that to understand the structure of that protein, the active site of that protein. The active site residue may be mutated due to the altered function, but still we can understand that how these active site residues might be functioning for the protein sequence in hand, and therefore the prediction of the three-dimensional structure using this homology modeling can be useful in understanding the functioning of the protein, such as an enzyme or a hormone receptor or a transcription factor for binding onto the DNA for regulation of gene expression. Therefore, in the present module on protein structure modeling, we'll attempt to obtain an experimental 3D structure from the protein database at RCSB. In case the experimental structure is not available, we'll learn obtaining a model of the three-dimensional structure. If a model is also not available or not adequate for the purpose at hand, then automated homology modeling will be undertaken to model the protein structure. Finally, we'll learn to judge the quality of the protein model so as to decide whether it can be used to meet the purpose at hand or not. The three-dimensional structure information of proteins is useful to derive enzyme interactions with its substrate for understanding catalytic mechanism and even for altering substrate specificity, as well as for exploring allosteric control of the enzyme activity through understanding interactions of the enzyme active site with allosteric binding site, binding its regulator. In addition, the three-dimensional structure information is useful for planning site-directed mutagenesis of enzymes for improving their functioning. Further, three-dimensional structure information is important in understanding the interaction of two proteins as in signal transduction or gene regulation. Above all, 3D structure information is essential for simulating protein ligand docking for structure-based design of new drugs or even improving existing drugs, inhibitors and activators. In the present module, we'll take the example of bifunctional enzyme HPR kinase phosphorylase, that is HPR-KP, from Enterococcus faecalis for modeling its protein structure. In the next module, we'll use the HPR-KP for docking its substrate ATP. HPR-KP is a bifunctional enzyme to add phosphate to serine 46 of HPR as kinase and to remove phosphate from serine 46 of phosphorylated HPR as phosphorylase. As kinase, HPR-KP accepts HPR and ATP as substrates to produce serine phosphorylated HPR and ADP. In vitro, HPR-KP can accept pyrophosphate as substrate for kinase reaction to produce serine phosphorylated HPR and inorganic phosphate. Fructose 1,6-bisphosphate is a positive regulator and inorganic phosphate is a negative regulator of the enzyme activity. However, only ATP dependent activity is stimulated and pyrophosphate dependent activity is not regulated in vitro. As phosphorylase, HPR-KP accepts serine phosphorylated HPR and inorganic phosphate as substrate to produce unphosphorylated HPR and pyrophosphate. Dear students, in gram-negative Neisseria, serine phosphorylated HPR is involved in cell adhesion. In gram-positive Listeria, serine phosphorylated HPR is involved in transcription. In addition, serine phosphorylated HPR in gram-positive bacteria is a paradigm of signal transduction, where serine phosphorylated HPR acts as a co-repressor for the regulation of gene expression of those genes which are involved in carbon metabolism. In this module, we are interested in the structure of HPR-KP from Enterococcus faecalis. Therefore, visit PDB and start entering HPR-KINASE in the search text box As you type in, the text box will suggest the recommended names such as UniProt, molecule name or ontology terms to guide you. The first suggestion for UniProt molecule name for HPR kinase phosphorylase matches our requirement. Therefore, simply click on the first suggestion or enter complete name HPR kinase phosphorylase and click Go button. This will display the search results as shown. The organism column shows that structure from Lactobacillus, Bacillus, Staphylococcus and Mycoplasma are available for HPR-Kp. However, no experimental structure is available for Enterococcus faecalis. The PDB search has shown that the structure of HPR-Kp from Enterococcus faecalis is not available in the database, whereas the sequence is available in hand. Therefore, the sequence of HPR-Kp can be used for modeling its protein structure. Unfortunately, the rate which at the new sequences are determined far exceed the rate at which the structures are determined. For example, as in May 2016, there were around approximately 118,000 structures in PDB, whereas the well-characterized sequences in SwissProt component of the UniProt knowledge base at XPazi server were found to be 550,000. And this gap is further widening due to the genome sequencing project, with the result that we have a Tremble translated EMBL database which contains around 63 million protein sequences. Therefore, the genome sequencing projects have widened the gap further. Consequently, we want to assign the function, and for assigning the function to a sequence, we need a structure. Unless until we have a structure, we will not be able to assign the correct or the exact function to a protein sequence. Although it is possible to some extent to assign a function on the basis of sequence alone, but still yet central paradigm of bioinformatics ensures that it is the sequence, structure and function relationship which shall be used, and therefore the three-dimensional structure of the protein molecule for the sequence in hand is an essential or a prerequisite to assign the function to this sequence. The central paradigm of bioinformatics considers that the sequence information in DNA, RNA and proteins represents the same knowledge and determines the molecular structure of actual transforming machines, that is proteins and RNAs, to interconvert or transform metabolites and therefore controls the phenotype of a biological system. Therefore, central paradigm of bioinformatics specifies that sequence information determines the protein structures, which in turn will give a specific biochemical function to finally express cellular phenotype through interaction of metabolic, signal and gene expression pathways. However, due to general difficulties in determining the experimental structures using experimental techniques, there is considerable interest in predicting the three-dimensional structure of a protein. This prediction, or so-called protein structure modeling, helps to establish the intimate relationship between the sequence and function through this structure prediction. Therefore, if there is a predictor structure which we call a model, then we can use it for assigning the function to the sequence in hand. However, the model which is to be predicted should be adequate model for testing the hypothesis to explain the experimental data. For example, let us say that same enzyme HPR-Kp. We are interested in identification of the active site residues. Let us say from the literature, we find that these are the 3-4 residues which are involved in the mechanism for catalysis. And in case we are forming a hypothesis that at the place of aspartate, if we mutate it to a glutamate, then the Km or the Vmax may change. So if this hypothesis is there, we must check it only if we are having available structure. Once we have a structure, we can mutate it in silico and then express the mutated enzyme and then use that mutated enzyme to obtain experimental data. If the experimental data is explained in the way we predicted or hypothesized, therefore that helps in elucidating the mechanism for the catalysis. So therefore the availability of the structure is very important. It is well known that the proteins from the diverse sources may exhibit differences in sequences, but still they have similar structure. Sometimes sequence similarity approximately 30-35% and above shows that they are descendant from a common ancestor. Once they have descended from a common ancestor, it means they will be having a common folding pattern. Folds, as I have already told, that there are finite number of folds available in the PDB structures. These are estimated to be around 3000 folds and they are not expected to increase much more. Whereas the structures available till May 2016 were 118,000. Therefore, the folding pattern being similar for the homologous proteins and due to the limited number of three-dimensional structure, three-dimensional folds, maybe due to steric or other clashes, we can presume that the two sequences in hand for which one sequence is having known structure and other is not having known structure, we can use this structure as template to assign this structure to this known sequence to be taken as target for assigning the structure. The modeling rationale therefore includes homologous proteins which have substantial sequence similarity due to descent from a common ancestor and are invariably found to have similar tertiary structures. These includes proteins with similar functions from different species such as sperm whale myoglobin and horse hemoglobin. These first ever two protein structures solved, despite being from widely different sources, are highly similar in sequence and 3D structures. Members of cytochrome family is another such example. One other category of homologous proteins shows sequence similarity in fold and has common function. In most such proteins, highly conserved residues are involved directly in the function or they are required for maintaining the structural integrity of the functional relevant region. The best example is the structural similarity between mammalian serine proteases such as chymotrypsin from mammals and bacterial serine proteases such as subtilisin. Only the residues which are involved in catalysis are identical between these two families. Common mononucleotide binding fold observed in many enzymes like lactate dehydrogenase and pyruvate kinase is another example. The third category of similar proteins corresponds to the observations of the structural similarity with no obvious sequence similarity. There is no functional similarity either. The most notable example is that of antibodies domain, that is immunoglobulin fold. Dear students, in the module 10, we have seen that the alignment between two sequences is meaningful to infer homology if there is sufficient identity between the two sequences. Therefore, the structural knowledge which is available for one sequence can be applied to predict the structure of the second sequence. Since we have not obtained the structure of HPRKP from Enterococcus faecalis in the PDB database, let us see online if a predicted structure of this HPRKP is available for Enterococcus faecalis, so that we are able to use that for computing or detecting the active site residues and for docking in the next module. Visit Protein Model Portal, which provides predicted protein structure models. In case the web interface does not show the search text box, click on the menu button to open search text box. Enter HPR kinase Enterococcus faecalis V583 in the search text box and press enter key. The search results page returns two entries. Click on Swissprot account number O07664 for HPRKP to reach the available model of this sequence. This shows that there is one model available based on 1KNX template. The model is predicted from template 1KNX having 27% identity to HPRKP sequence from Enterococcus faecalis. The accuracy of the predicted model strongly depends on the sequence similarity between the target protein and the template. If more than 30% sequence identity is found among the target and the template sequence, then the backbone of the model may be as good as a crystallographic structure. However, large errors are expected if the sequence identity falls below 30%. In addition, the accuracy of the predicted model also depends on the mode of predictive modeling. If automated mode without manual intervention is used to predict structure following homology modeling approach, then at least 30% sequence identity between the target and the template is essential. The model is predicted by modeler with only 27% identity for the complete chain of HPRKP. Click on Modbase hyperlink to reach Modbase model information page. Open the drop-down list, perform action on this model and select coordinate file option to download the structure in PDB format. Select Swiss PDB viewer as program installed on your local computer to open this file. Now save the structure on the local disk from within Swiss PDB viewer menu interface, opening file menu, then selecting save command and choosing current layer command to save the structure. The principle behind the homology modeling is based on the fact that the structures of the homologous proteins are better conserved during evolution than the conservation of their sequences. Therefore, it has been found that the proteins, even with the dissimilar sequences, may assume or acquire the similar three-dimensional structures. There are two main assumptions for the homology modeling. The first is that those residues which are similar or same in the two proteins, that is the template and the target, the 3D coordinates are assigned exactly between these two template and target sequences are simply borrowed. So it is presumed that they will be having similar spatial coordinates. Second assumption is that if there are mutations or insertion deletions, particularly insertions and deletions, if they are mainly in the loop regions. In the protein structure hierarchy module number 11, we have seen that the core of the protein is tightly packed and the active site is located in a cleft which is present on the surface and is formed by the loops. Therefore, insertions and deletions are not presumed to be occurring within the secondary structures, regular secondary structures, but are presumed to be occurring within the loops which are not repeating not having repeating phi and psi angles. Therefore, we can say or presume here that the two assumptions are to be followed. Now, second is the extent of sequence identity. The model which we have seen at Modbase predicted by modeler is having only 27% sequence identity between target and template K and X. Therefore, there is a need to take a careful judgment or evaluation of the quality of that model before it is being used for detecting the active site or constructing hypotheses. Even otherwise also, there shall be a fresh effort for modeling the HPRKP from Enterococcus faecalis because the structure, the new template might have become available in the meantime. So it is always better to go for fresh modeling at same PMP protein model portal, so that in case a new template has become available, the same may be used. And also the two models, one already predicted and one achieved through fresh modeling, can be compared for judging the quality of the two models, and whichever comes to be the best can be used for the meeting the purpose at hand. So we need to have an adequate model which can explain the functioning in which we are interested. At PMP server, user can submit a new modeling request to various homology modeling servers. Let us model UniProt account 07664 for HPRKP using Swiss model. Reach PMP server, enter the requisite details, choose Swiss model only and click Submit Query. The resulting page will show the status and a link to Swiss model results page. The link for the predicted models will also be received via email. User needs to register at Swiss model with a valid email ID and a chosen password. The request submitted for Swiss model server uses automated mode for homology modeling. The results page lists all the models predicted. Each model has 4 panes. The top left pane of the model shows the general information about the template used and the model quality parameters. The bottom left pane of the model shows structural sequence alignment. The top right model of the shows that a model is shown in secondary structure ribbon form, and the bottom right pane shows the coverage of alignment of the target and the template residues used for homology modeling. The top left pane of a model shows that global model quality estimation, that is GMQE, is 0.75 and Q mean is an acceptable thumb. The GMQE may vary from 0 to 1. Higher the GMQE, the better the model quality. The sequence identity for model 1 is approximately 50%. with almost full alignment coverage. The template chosen for the first model is 1K07, which is different from 1KNX used for modeling by modeler at Modbase in 2012. This fresh modeling was undertaken in 2016. At Swiss model, the automated mode is suited for cases where target template similarity is sufficiently high to allow for fully automated modeling. As a rule of thumb, automated sequence alignments are sufficiently reliable when the target and the template share approximately 50% sequence identity or higher over the aligned residues. Let us save model 1 as PDB file on the local disk with file name model_1K07a.pdb by opening the drop-down list for model 1. The bottom left pane of the model shows alignment, sequence alignment. Click on the alignment formatting options tool displayed at the beginning of the alignment to open alignment options. Check the checkbox to fade mismatches in the alignment. The other available options for formatting alignment are for choosing a secondary structure format DSSP Cypred or SSPro. For the present example, choose Cypred. In addition, user can choose a color scheme. Choose the color scheme for the structure. There is an option to export alignment in FASTA and cluster formats. After setting the options as shown in this slide, just click anywhere in the alignment. The alignment highlights secondary structure in Cypred format with strands as green and helices as light blue. The model template alignment shows that 5 out of 8 residues in the P loop, which is highlighted in this slide in the rectangular box, are not present in any regular secondary structure. The P loop is present on the surface and is involved in the binding of the substrate ATP. Therefore, the alignment of P loop on the surface is in agreement with the presence of binding site present on the surface in a loop. Model structure view displays the secondary structure in cypred format with strands as green and helices as light blue. Below the model structure view, the bottom right pane shows the length of the alignment between the target and the template used for modeling target sequence for each of the predicted models. There are 6 models predicted. Template is shown in green and the target in each of the predicted model is shown in light blue. A red colored border bar highlights the currently displayed structure in the top right pane. Take the mouse over the first model bar to display the coverage of alignment length. The first model has alignment coverage from 5 to 302 residues. Now move the mouse over the sixth model to display its alignment coverage, and the bordered bar will display highlighted in blue border that the sixth model has alignment coverage length from 68 to 132 residues. Each vertical bar in this bottom right pane has a sequence length of 10 residues, starting from first with 1 to 10, then second from 11 to 20, and so on. The sixth model alignment starts in the 7th column at 68 and ends in 14th column at 132. In future, users can visit Swiss Model Workspace and choose My Workspace in Modeling tab to view any of the previous modeling projects. Scroll down the workspace for previous modeling projects and click on the project to view model results. Model 01 and Model 06 has acceptable Q mean values of -2.04 and -2.06 with a thumbs up image, and the rest are unacceptable with a thumbs down image. However, Model 6 does not cover the residues in P loop from residue 155 to residue 163. Therefore, there is no need to proceed with Model 6. But with Model 1, we need to judge the quality before using it to arrive at a conclusion that it is adequate to meet the purpose in hand. Dear students, before judging the quality of a model, we need to keep in mind that a model is never correct or incorrect. It is either adequate to meet the purpose in hand or it is inadequate. For example, if you look back, the model of the atom, that the atom is indivisible, but was not able to explain when it could be divided between positive charge and negative charge with the discovery of the electron. Therefore, a plum pudding model was developed. And in the plum pudding, all the sphere was considered to be positively charged, and small electrons were placed on this to give a plum pudding model. But then with the Rutherford's experiment of alpha particles striking on to the metal sheet, showing that the positive charge is centered around and rest of the space is empty. So this gave a nuclear model, as in the solar system, sun and earth are revolving around the sun and electrons revolving. So as and when the new experimental data is not explained by the model in hand, the model needs to be refined. Therefore, a model is never correct or incorrect. It is either adequate to explain the experimental data or to understand the hypothesis can be explained or not. Therefore, for the present example, for the model of HPR-KP, we'll keep in mind the experimental information known about the HPR-KP. The HPR-KP has a Walker-A motif known as P loop, which is involved in the binding of a ATP. In other proteins. Therefore, this information is very useful to understand that whether the model is adequate or not. So therefore, to get adequate or meaningful biochemical insight for the mechanism of enzyme through P-loop, we need to understand this only, whether the P-loop is correctly oriented or not in the modeling. The present, we are having now two models. One was predicted at Modbase by modeler using the template 1KNX, and the second was obtained through fresh modeling using Swiss model with the template 1KO7. The quality of a model is checked by comparing its structural parameters with those derived from crystallography solved template structures. Both Swiss PDB viewer and Swiss model can be used to assess the quality of a model. So we will be using both Swiss PDB viewer and Swiss model to assess the quality of these two models, one available at Modbase predicted by modeler and other obtained through fresh modeling using Swiss model. Swiss PDB viewer can be used to inspect the quality of the model qualitatively as well as quantitatively. Using B factor and manual visual inspection, it allows visual comparison of the two structures qualitatively. It also allows to quantify structural differences by measuring root mean square deviation between two structures. Swiss PDB viewer can detect amino acid residues making clashes in a protein structures, and Swiss PDB viewer provides to view Ramachandran plot to view torsion angles for selected residues. Ramachandran plot allows detecting those residues which fall outside the allowed region. To appreciate structure quality of the model, we need the model and the template structure. We have already saved the model 1 as PDB file on the local disk with the file name model_1q7a.pdb. Visit RCSB PDB and download 1q7 template structure in PDB format. Open Download Files tab in PDB. Choose PDB command. This will open a dialog box to open with or save as or save file dialog box. Open drop down list to open with and choose command other. This will open a dialog box to choose helper application to open the file 1q7.pdb. Click browse button and select file spdb.exe stored on your local disk. This will prompt with opening 1q7.pdb dialog box. Click OK button. This will open the structure file using Swiss PDB viewer. In the control panel of Swiss PDB viewer, select all the residues of chain A. To select, click on the first residue, then browse to the last residue in chain A. Here, press Shift key on your keyboard and click the last residue, that is ASN298. All the residues in chain A will be selected and will be shown now in red. Now open File menu and mouse over Save command to open subsequent menu. Choose Selected Residues of Current Layer command and save with file name 1ko7_a.pdb file name. Now close the template file 1ko7. Follow the stepwise criteria for judging the quality of the models. That is, open the saved file, template file 1ko7_a, and next open predicted structure of HPRKP, which was saved as model 1ko7a. From the display menu, set display to stereo view in case you are not in the stereo view. Models can be evaluated visually using B factor coloring and qualitative inspection. With the two structure files open, color both the structures as B factor from color menu. The B factor describes the displacement of the atomic positions from an average, that is mean position value. For example, more flexible an atom is, larger will be the displacement from the mean position. In graphic programs, user can often color a protein according to B factor value. Usually the regions with low B factor values are colored blue, that is less displacement, while high B factor colored red, that is more displacement. Therefore, blue to green regions are acceptable and red regions in the model are not surely correct structures. In the model structure, there are no red regions. Therefore, model structure may be presumed to be correct structure for the purpose, or may be presumed to be adequate structure for the purpose in hand. Now press Control key with your thumb, left thumb on your keyboard, and click Tab key. This will swap the structure from template to target. Again press Tab key without leaving the Control key. This will again swap the structure. So go on. In a quick fashion, you will find template structure, target structure, template, target. In this way, swapping the structure, you will be able to make out manually, visually, that what are the residues which are occupying the same positions in the three dimensions, and which are the residues which are occupying different positions in template and target. You will realize that the core structure is having the residues placed in both these model and the template at almost similar positions, whereas the surface residues, they are occupying slightly different positions. Now set the color of the model as cyan color in the 6th column of the control panel. Now set the color of the template as using type command in the color menu. P loop residues extend from 155 to 162 in the model and from residues 151 to 158 in the template. Select these residues in both the structures from the control panel and make these visible by marking V in the first and the second column of the control panel in each of the structure layer. Now center the view. Now press Control key and click Tab key to view template. Now press Control key and click Tab key to view the model. Repeat clicking Tab key again and again while the Control key is always kept pressed to view one structure at a time, and this clicking Tab key will swap the structures to appreciate the difference between the two structures. You will find that P loop residues in both the structures are having almost identical positions. With the two structures open and P loop residues selected currently, select Calculate RMS command from the Fit menu. Set the options in the ensuing RMS and AutoFit options dialog box for using all atoms and 1KO7_A layer as a reference, that is fixed layer, and model_1KO7A as second layer. Click OK button. This displays the number of bonded atoms involved as 58 and calculated RMS equal to 0.07 angstroms below the toolbar, that is displayed below the toolbar. Therefore, P loop in the model is occupying almost identical position in both the model structure and the template structure. Similarly, follow the procedure to extract template chain F from template file 1knx.pdb and save as 1knx_f.pd. Follow the stepwise criteria for judging the quality of the mod-based model. Follow same steps using 1knx template and model files. The only difference is that while calculating RMS, select only 8 matching residues in both the structures. The calculated RMS value of 3.58 angstroms for mod-based model is much higher than 0.07 angstroms for Swiss model predicted during fresh modeling. Therefore, for designing drugs against Enterococcus faecalis HPR-KP, Swiss model predicted structure can be considered to be adequate enough to be used for docking inhibitors in the P-loop active site of the HPR-KP enzyme. Make model as the active layer from the control panel and detect amino acids making clashes from the select menu and choose command residues making clashes. Similarly, make template as the active layer from the control panel and detect amino acids making clashes from select menu by choosing command residues making clashes. Now from the wind menu, choose command layers info to open layers info window. The result shows that the predicted model does not have any residue making clashes. From the color menu, select first command so that all subsequent commands act on both backbone and side chain atoms. Now from the color menu, choose command protein problems. This will communicate the coloring of residues which are in problem in the HPR-KP model. Amino acid side chains buried without hydrogen bonds will be colored orange. Scroll down to the P-loop residues in the control panel. The gray color for each of the P-loop residues shows that none of the active site residue in the P-loop is in any problem. With the P-loop residue selected as above, open wind menu and choose Ramachandran plot command. This will display the Ramachandran plot for the selected P-loop residues. There are three regions in the Ramachandran plot. The first is the yellow region, which displays that all those residues which have favorable or allowed torsion angles, that is the region enclosed within a yellow line. Outside this yellow line is the region marked with a next boundary in the blue color, which shows that the residues having phi and psi angle in this region are allowed residues to have less favorable torsion angles. Outside this blue boundary is the area which is disallowed to have torsion angles except for glycine. In this area, only glycine is allowed to have these torsion angles. Take the mouse over each of the squares present in this disallowed region, that is not within blue and yellow regions in the plot. The labels for the residue will be displayed on the top left corner. These are shown to be glycines. Therefore, the predicted model underscore 1K07A structure has all the amino acid residues with allowed torsion angles either in the favored region or allowed and glycines in the disallowed. Using Swiss model tools, we can estimate local model quality using Anolia and Gromos plots, as well as evaluate secondary structure features, geometrical features, and solvent exposure along with stereochemical check. Visit Swiss model for using model assessment tools and select Anolia and Gromos for local model quality estimation. Select ProCheck for stereochemical quality estimation and choose DSSP for secondary structure definitions. We have already seen that the Q-mean and the global model quality estimation values for the predicted model are acceptable. The y-axis of the Anolia and Gromos plots represents the energy for each of the amino acid of the protein chain. Negative energy values shown in green represent favorable energy environment, whereas positive values which are shown in red are not favorable energy environment for a given amino acid. The 8 residues in the P loop are highlighted with a black rectangle. The DSSP code for first residues conveys that it is an end of an extended sheet participating in a beta ladder. This is followed by 5 residues which fall in bands and hydrogen bonded turns, which are loops on the surface of an enzyme. The last 2 residues form part of the helix to enter the core of the enzyme. From our knowledge of the protein folding, we know that regular secondary structures are buried inside the core, and the loops, which usually harbor active site or binding site, fall on the surface collapse. The Anolia plot is perfect for these residues, whereas Gromos plot for 4 residues, that is serine, glycine, isoleucine and glycine, that is from 3rd to 6th residues, show slightly unfavorable energy pattern during folding. Dear students, it has been estimated that energetic criteria alone is unable to distinguish correctly folded structures from the incorrect ones. This is because some of the incorrectly folded proteins are capable to reach the same energy minima as of the correctly folded ones. Hence, the empirical comparison of parameters such as torsion angles, bond lengths, etc. between a model and its template structure may be used. Consequently, let us use ProCheck pattern for evaluating the stereochemical quality. The ProCheck suite of programs assess the stereochemical quality of a given protein structure after comparison with the stereochemical parameters derived from the well-defined high-resolution structures in the database. The ProCheck report shows nearly 90% residues are present in the core, correct, and nearly 10% are having torsion angles with allowed phi and psi values. Only 1% residues are present in the disallowed areas, which we have already seen are mostly glycines. All this in always shows that the model has favorable stereochemical quality. Therefore, the predicted model at Swiss model workspace is adequate to take docking of an inhibitor or a substrate such as ATP in the P loop of the active site of the HPRKP from Enterococcus faecalis for designing a drug against this pathogen. We have seen in this module that when an experimental three-dimensional structure is not available, we can search for a predicted model of the protein in hand. And we found that yes, there is an available protein model for HPRKP at protein model portal. This was predicted with modeler at the mod base. However, we find that instead of accepting already predicted model, we shall go for fresh modeling because due to the reason mainly that a new template might have become available. And once we take fresh modeling, then in that case we arrived at 2 models, one already predicted, one fresh predicted using Swiss model. Then the job was to estimate or to judge the quality of both the models and to choose the best one. And we evaluated the quality of the predicted models using Swiss PDB viewer and Swiss model. Using Swiss PDB viewer, the structure quality was evaluated through visual appreciation using B factor coloring and superposition comparison. The deviation in the structure of the model and the template was quantified using root mean squared deviation calculation. We also could detect using Swiss PDB viewer the amino acids which might be making clashes in the predicted model, or even could detect the protein problems. So both were explained in this module. The Anolia and the Gromos plots were used to appreciate or to evaluate the local model quality. The stereochemical check was also performed using Ramachandran plot and ProCheck software. Therefore, we can, with all these judging and evaluating criteria, we could arrive at a conclusion that the model which is predicted afresh using 1K07 as template with 50% sequence identity in the alignment was found to be a better model than the one already available at PMP with the 27% sequence identity in between the alignments and predicted using 1KNX. Therefore, the fresh model predicted with 1K07 can be presumed to be adequate for undertaking the docking of, say, an inhibitor so that the HPRKP is inhibited, and that inhibitor can later on be used as a lead compound to develop a drug against HPRKP of Enterococcus faecalis pathogen. I thank you all for visiting EPG Pathshala.